Ladies and gentlemen, welcome to discussion, this lecture for cell anatomy and physiology, focusing on the cell membrane. In terms of cell membrane function, we will cover a number of things, including the fact that all cells are surrounded by plasma membranes, and because this is important, the cell membrane then becomes the without the cell membrane, then there's no cell, because that's our barrier, that's what defines the cell. Cell membranes are composed of a lipid, a double layer or bilayer, or phospholipids that are mixed or embedded with globular proteins, such that on the external side of the cell membrane we have carbohydrate molecules that join with lipids and form glycolipids, and those that combine with proteins, they form glycoproteins, and these glycoproteins and glycolipids have specific physiological functions that they provide to the cell as identity markers. So basically, this is what we are talking about in the picture. We've got the cell membrane, which is made up of molecules of phospholipids, one on the inner side of the cell, one on the outer side of the cell. So the phosphate is facing on the outside towards the water, and on the inside towards the water intracellularly. And then in between, we've got the tails of the phospholipids that make the interior of the cell membrane fatty. And then we've talked about the fact that embedded within these two layers of phospholipids are the proteins. Okay? And then we also have proteins that complex with carbohydrates, they form glycoproteins, and the lipids that complex with the carbohydrates form glycolipids. So in 1972, S. Singer and G. Nicholson proposed the fluid mosaic model as the approach to discussing the structure of the cell membrane. And so these two, they proposed that the cell membrane should be seen as a double layer of phospholipids that slide on top and below each other. So they slide across each other. So meaning the membrane is fluid, it's able to flow one layer on the other. Then mosaic in relation to the fact that the phospholipid bilayer, there is a mix of proteins that make part of the structure. Therefore, it is referred to as mosaic. Then we continue with the structure of the cell membrane. So we talked about that the phospholipids, they are chemically made up of glycerol and fatty acids and the phosphate group. Okay, so the glycerol and the two fatty acid tails, they make up the fatty part of the cell of the phospholipid, and then the phosphate head is the polar side of the molecule. So the phospholipid bilayer, mainly two layers, phospholipids, as we have said, which is structured in such a way that the head is polar and the two tails are non-polar. So it also contains, the bilayer also contains cholesterol. As in animal cells, cholesterol is very, very important. We're going to see how it improves fluidity and the ability to slide for the two layers of phospholipids. So we have talked about that the bilayer is fluid, allowing proteins to move around within the bilayer, and also the two layers sliding one on the other. So this is what we are talking about. The heads of the phospholipids are oriented towards the water side extracellular and water side intracellular, and then the tails are oriented towards each other, making the interior of the cell membrane non-polar hydrophobic, meaning water hating, meaning they do not have the affinity for water. Then polar hydrophilic heads, meaning the heads of the phospholipid molecules on either side of the membrane, they are of affinity for water. The cholesterol we've talked about, that is part of the core of the cell membrane, affects the membrane's fluidity within the animal cell. Therefore, cholesterol is not a bad molecule to the animal. It's excess cholesterol that is bad and forms hypercholesterolemia related problems of clogging of blood vessels. Otherwise, cholesterol, as you will see here, interdigitates amongst the tails of the bilayer, making the tails and the bilayer more fluid and be able to slide one on the other. The membrane has proteins, and these proteins have got functions. We're going to go through that. But also a membrane as a controller of movement of substances. We'll talk about this in the next lecture. And we'll be talking about all the passive and active processes that ensure that materials are transported into and out of the cells. So membrane proteins, quickly we look at membrane proteins. These, if you still look at the membrane as described using the fluid mosaic model, we see that the membrane is a collage of different proteins embedded in the fluid matrix or the fluid bilayer. And these proteins, some of them are on the surfaces of the bilayer, so therefore they are called peripheral proteins, and they are loosely bound to the surface of the membrane. And we can see in this structure where on the surfaces there are these proteins associated with the membrane. But we also have proteins that are called integral proteins, and these proteins, they penetrate the hydrophobic core of the bilayer. So they enter the interior of the bilayer, and they could actually either be partially embedded, meaning in one layer, or throughout both layers. If it's throughout both layers, they are called transmembrane proteins. Otherwise, transmembrane proteins are integral proteins, but not all integral proteins are transmembrane proteins. So this is what we are saying. So for example, this is a transmembrane because it has traversed the both layers or spanning the bilayer. So what are the functions of cell membranes? Cell membranes regulate the passage of substances between the outside and the inside, and the inside and the outside. And the membranes are important for detecting of chemical messengers arriving on the surface that can actually affect the cell in terms of function. The cells also link adjacent cells to each other through what are called membrane junctions. So these membrane junctions are made up of what? Membrane proteins. Then we also have membrane proteins forming anchors of cells to the extracellular matrix. So then we look at the membrane proteins specifically so that we can see how these membrane proteins contribute to the totality of the function of the cell membrane. So 6 major functions membranes perform as transport structures within the cell membrane. We also have membrane proteins performing enzymatic activity, where proteins that make up the cell membrane may be enzyme in nature, leading to their ability of these proteins to convert specific substances into products that become useful in the cell. For example, some of the membrane proteins like sodium potassium ATPase converts ATP into ADP and inorganic phosphate to produce energy that drives active processes. Signal transduction, a membrane protein may have a binding site, okay, with a specific shape that fits a particular chemical messenger or a hormone. Then the external messenger or signal may cause a conformation change in this protein that relays a message into the cell and cause physiological changes. So basically, the illustrations is what we can see in the picture. And then we also have the others where cell membrane proteins become cell-to-cell recognition structures, where some glycoproteins act as identifier tags that are specifically important to recognize other cell types. For example, white blood cells for your body will actually be identifying cells of that particular body by specific glycoproteins. So that is why you find that someone cannot receive a wrong blood type, because the immune system for that person will actually not be able to recognize those, and it will attack them and cause hemolysis. The membrane proteins also work as intercellular joining structures. They also work as attachment structures for the cytoskeleton and the extracellular matrix. So myofilaments or other elements of the cytoskeleton can be used to bond the membrane proteins, and this can function to help maintain cell shape and stabilization for the location of certain membrane proteins within that. So proteins that are there to the extracellular matrix can coordinate extracellular and intracellular changes. So this is very, very important. So what we've been able to discuss is the cell membrane in terms of structure, its anatomy, and how the cell membrane can be described using the fluid mosaic model, and how cholesterol proteins are part of that, and we have looked at their functions. Look out for the next part of the lecture on the cell membrane. Thank you.